Hello, thank you for taking the time to view my talk. I'd like to thank the conference organizers for giving me this opportunity. My name is Aaron Blanchard, and I'm a fifth-year PhD student in the laboratory of Dr. Khalid, shown here, where, among other things, we develop DNA mechanotechnology for studying, controlling, and generating piconewton scale mechanical forces. The ability to image biomolecules in living cells has become central to biological research. Thanks largely to fluorescent imaging tools, our ability to understand and manipulate biomolecular processes has allowed us to see cells not only as chemical systems, but also as small mechanical systems that interact with their surroundings by generating and responding to mechanical forces. And because mechanical forces are important in biology, we naturally have a lot of questions about them. What roles do they play? How do biomolecules respond to force? What are the properties, such as magnitude and duration, of these mechanical forces? But forces are transient and intangible. You can't tag them with a fluorescent protein the way that you would tag a biomolecule. Accordingly, we have to come up with clever methods for visualizing biomolecular forces in living cells. Our goal in the Salida lab is to bring tools and techniques for imaging molecular scale forces to the biophysics community. The most common tools for visualizing cellular forces make use of deformable substrates on top of which cells are cultured. In the example shown here, called traction force microscopy, cellular forces cause substrate deformation. Optical microscopy is then used to track the displacement of beads embedded within the substrate, and computational processing is used to back-calculate cellular force. While such techniques have been incredibly useful, they offer micron-scale spatial resolution and report on the combined traction forces of the entire cell, rather than the forces of individual molecules. To address this concern, we and others have developed molecular tension probes. There are many different types of molecular tension probes, but they all have the same key components: a fluorophore and a quencher separated by an extendable domain. Generally, these probes are anchored to a planar surface such as a glass cover slip, and then present a ligand that can bind to a cell receptor. We functionalize these probes at high density onto a surface and then culture cells on top of them. The cell's forces pull open the probes, dequenching the fluorophores and allowing us to visualize force with fluorescence microscopy. The most precise molecular tension probes are DNA hairpin-based probes shown here. In our design, a DNA hairpin is flanked by a fluorophore quencher pair. Force exceeding a sequence-dependent tension threshold, called the F1/2, opens the hairpin and results in a 30 to 100-fold increase in fluorescence intensity. DNA hairpins have very tight force response thresholds, making these probes essentially digital switches that offer very precise quantification of molecular force magnitudes. These probes also offer at least an order of magnitude improvement in spatial and temporal resolution when compared to traction force microscopy, and signal is generated from the mechanics of individual receptors. These properties make DNA hairpin-based tension probes very useful for characterizing the fundamental properties of molecular forces. This is a video of a platelet spreading on a DNA hairpin functionalized glass cover slip. RICM allows us to see where the platelet is interacting with the cover slip, and our fluorescence image shows a map of integrin tension. Bright signal indicates a region where a large number of tension probes are opened by this cell. We can see that after a few minutes, the platelet has begun to spread on the surface and open some tension probes. Eventually, the platelet spreads fully onto the surface and is pulling probes open in two distinct regions: a lamellopodial ring at the outer edge and a strong inner mass of tension in the middle. These features are impossible to see with substrate deformation techniques such as traction force microscopy, because the resolution of those techniques is similar to the length scale of the platelet itself. One limitation of our technique is that, while it precisely reports on tension magnitude, it does not report on force orientation. However, research has increasingly shown that the orientation of mechanical force is important in many biological processes. For example, integrin receptors, which are generally known to anchor mammalian cells to the extracellular matrix, are known to be mechanosensors. From our current understanding, force transmitted through an integrin separates the protein's two units, exposing binding sites that can initiate important pro-growth signaling pathways. Molecular dynamics simulations from the Tim Springer lab have suggested that tension applied laterally to the plasma membrane promotes separation of these two domains, while force applied vertically does not. This model, which has been supported by literature findings, suggests that integrins act as orientation-dependent force sensors. And while I'm not showing any data on this today, recent work has suggested that the T-cell receptor, which uses force to detect foreign antigens as part of the mammalian immune response, is also preferentially activated by lateral tension. While literature findings have generally supported these lateral activation models, our ability to experimentally test these hypotheses has generally been limited because we do not have tools for directly measuring the orientations of receptor forces in living cells. To address this limitation, we developed a method that uses fluorescence polarization microscopy to measure force orientation. Fluorescence polarization techniques hinge on the fact that fluorophore excitation depends on how well the excitation beam's electric field is aligned with the fluorophore's transition dipole moment, which can be approximated as a 3D vector. Our DNA hairpin probes utilize a fluorophore called cyanine 3b. When you conjugate a cyanine dye to the terminus of a DNA duplex, as is the case in our probes, the fluorophore stacks perpendicular to the duplex's long axis due to hydrophobic interactions. Therefore, a fluorophore in an opened tension probe will be perpendicular to the force orientation, as shown here. Now, we assume that an open tension probe can freely rotate around its long axis. And so if you integrate over time, this transition dipole moment actually becomes a disk that's perpendicular to the force orientation. If you use linearly polarized excitation light, then the electric field can be represented by a dipole, as shown in cyan here. The strength of excitation is going to be related to how well the fluorophore disk and the electric field dipole are aligned. Because the electric field dipole is restricted to the xy plane, you can get a sense of this by projecting the disk down onto the xy plane. The projection takes the shape of an ellipse. When the electric field is aligned with the long axis of the ellipse, the excitation is strongest, and so you'll achieve maximum fluorescence intensity. When the electric field is oriented with the short axis of the ellipse, you get minimal fluorescence intensity. As you can see here, the xy projection of the force runs right through the short axis of the ellipse. This means that if you can rotate the electric field and find the angle that minimizes your fluorescence intensity, you can measure the in-plane angle of the force. Furthermore, the shape of the ellipse is related to the tilt angle of the force. If you point this thing straight up, the projection of the disk is just a disk. But the more you tilt the force over, the narrower the short axis of the ellipse becomes. Therefore, if you can measure the ratio between the maximum and the minimum intensity values, you can also estimate the tilt angle of the force. To perform these measurements, we implemented a half-wave plate mounted in a rotatable actuator into the light path of our excitation laser beam, which is linearly polarized to begin with. A half-wave plate rotates the angle of linearly polarized light, and so this allows us to fully rotate the electric field vector within the xy plane. We used this setup to image our tension probe samples several times with different electric field angles. In total, we used 73 different polarization angles, and the entire acquisition took roughly 3.5 seconds. An example image of a platelet tension of a platelet's tension is shown here. For each bright pixel in the image, we fit the intensity versus electric field angle data to a sinusoid. Finally, we calculate force orientation from the features of the sinusoid. To give you an example of how orientation is related to the fit sinusoid, I prepared this animation. As you can see, as the force orientation is rotated around the z-axis, the fluorophore projection ellipse rotates and the sinusoid phase shifts. As the tilt angle of the force changes, the aspect ratio of the ellipse changes, resulting in modulation of the amplitude of the sinusoid. Extracting force orientation in a pixel-by-pixel manner allows us to map force orientation with diffraction-limited resolution. Here, we can see a force orientation map where every bright pixel has a dipole associated with it. The orientation of the dipole denotes the measured in-plane angle of the force, while the color of each dipole denotes the tilt angle. As before, we can see that the platelet has two distinct zones of tension that, interestingly, appear to exhibit two distinct patterns of force orientation. The lamellipodial edge exhibits forces that are oriented primarily towards the center of the platelet, and the inner region, on the other hand, exhibits forces that are directed primarily towards the central axis. You can read more about this technique, which we have named molecular force microscopy at Nature Methods. We have also used MFM to characterize integrin forces in fibroblasts. In this case, we can see that forces are concentrated to focal adhesion structures that point into the cell centroid. Interestingly, we have found that platelet and fibroblast integrins both exert forces with tilt angles of roughly 45 degrees, which is consistent with the lateral activation model that I discussed earlier. However, we notice a key difference between tilt angles between the two cell types. While platelet integrin forces had the same 45-degree tilt angle throughout the cell, fibroblasts exhibited a gradient in tilt angles, with forces closer to the cell centroid pointed more vertically than forces at the edge. We also imaged platelet aggregates, and we were able to distinguish forces exerted by single platelets within these aggregates, which would be impossible with substrate deformation-based techniques. Once we developed the technique, we started thinking of ways to make it more accessible to biological researchers. We found the limitation of our approach is that it's impossible to modify many commercially available microscopes to implement MFM. Our microscope scheme is pretty simple, and the addition of a rotational actuator should be pretty trivial to people who have their microscopes built out on an optical bench. However, a fair amount of fluorescence microscopy is conducted with commercial systems, many of which do not have free space laser lines that components can be easily placed into. Just to give an example, here's a really common microscope, the Nikon Eclipse TI, and the fiber optic cable that delivers the excitation laser lines. The excitation light plugs directly into the microscope here. Accordingly, it's not possible to install a rotatable half-wave plate into the excitation path of this microscope. Therefore, we knew that many researchers wouldn't be able to implement MFM. So we started looking for accessible, alternative imaging modalities that would enable implementation of MFM in a turnkey fashion. And by happenstance, we found a recent paper in Nature Communications from the lab of Peng Xi at Peking University in China that showed that you could use a structured illumination microscope to perform the same type of fluorophore orientation mapping that molecular force microscopy requires. Structured illumination microscopy, or SIM, is a super-resolution imaging technique wherein diffraction grating is used to generate an excitation pattern that is sinusoidal rather than flat. The diffraction pattern is then shifted and rotated while multiple images of a sample are acquired. These images are then fed into an image reconstruction algorithm that can double your spatial resolution. Importantly, in order to create the illumination pattern, the light must be linearly polarized with an electric field vector orientation that is parallel to the stripe pattern. Therefore, when the diffraction grating is rotated, the polarization is rotated along with it. So it turns out that any SIM microscope can be used to map the fluorophore orientation. And you can still use a super resolution reconstruction of SIM in order to get super resolution fluorophore orientation maps. Here's one of their example images where they've mapped the orientation of actin tubules. Here they're depicting fluorophore orientation in a different way than we did, by using color to denote the in-plane angle of the dipole and brightness to depict the maximum intensity. It turns out that SIM microscopes are pretty common, and there's a good chance that your institution has one. We actually have two at Emory, both of which are maintained by our Integrated Cellular Imaging Core. And so when this paper came out, we were eager to try it out with our tension probes and see if we could use one of Emory's SIM microscopes to create super resolution. So we put platelets on our tension probe functionalized surfaces as before and imaged them using a conventional SIM microscope. Here's an example acquisition. A total of 15 images acquired over the course of about 1 second are shown. Each row has a different orientation of the illumination pattern depicted on the left, and each column has a different phase shift. However, instead of running these images through a typical SIM reconstruction algorithm, we started by just taking row-wise averages of these images to obtain 3 images, one corresponding to each of the 3 distinct electric field orientations. If you run a simple mathematical reconstruction to obtain the pixel-by-pixel best fit sinusoid, and then display the XY plane angle of the force in each pixel, you can see that the platelet is contracting radially inwards as we saw before. I want to note that I've switched to the color-based method for displaying force in plane orientation. If you reference this color wheel, you can see that the left and right sides of the platelets are red, meaning that they're pulling side to side, while the bottom and top regions of the platelet are green, meaning that they're pulling up and down. That's what we expected, so this implementation of MFM on a SIM microscope appears to have been successful. This acquisition is roughly threefold faster than our previous implementation of SIM, as it can be completed in roughly 1.2 seconds. So here's a bigger view of that same platelet, shown in both wide field and super resolution. We were able to run these images through a SIM reconstruction algorithm developed by the Shi lab to obtain a super resolution tension image and a super resolution force orientation map. We're trying to be careful about how we analyze the super resolution data because it's known that SIM reconstructions can introduce structural artifacts. But we do want to note that if you look at a simple, well-defined structure within the cell, such as the lamellipodial ring on the bottom left, you can clearly see a narrow two-fold resolution enhancement. So I'm about to wrap things up, but I just want to end by giving you a brief example of how MFM can be applied to accessing otherwise hidden information. SIM-MFM is a really versatile technique that can be implemented without substantial photobleaching, so it was easy for us to run a time-lapse of a platelet spreading on a surface. We then quantified a few different features of the platelet over time. We can see that the spread area and total molecular tension intensity both plateau within 4 minutes, meaning that the platelet has essentially finished spreading. However, if you take the circular variance of the in-plane force angles, then you see a surprising result. Circular variance is a metric for how the forces are organized. When circular variance is high, force orientations are evenly distributed, which in this context mean that the platelet forces are all pointing radially inwards towards the center of the cell. However, when circular variance is low, that means that forces are all pointing along the same direction. Here we see that the circular variance is decreasing linearly with time, even after the spread area and total raw intensity have both plateaued. This means that over the course of time, the platelet is transitioning from a mechanical state where it is isotropically contractile to a state where it is anisotropically contracting along an axis. You wouldn't be able to see this phenomenon without force orientation mapping, but it has important biological implications for wound closure and platelet function. So to summarize my talk, DNA hairpins are the most sensitive force sensors that have ever been reported. Molecular force microscopy enables visualization of molecular force orientation. Molecular force microscopy can be implemented on any SIM microscope, and SIM-MFM enables super resolution mapping of force orientation. With that, I'd like to thank the Salida Lab. I'd like to thank you for attending my talk. Thank you to Joshua Brockman, who was the co-first author on the original Nature Methods paper, and Alexa Mathesis, who we collaborated with throughout this entire project, who was at University of Alabama, Birmingham. I'd like to encourage you to please follow me on Twitter. My handle is DNA_mectech. And I'd also like to refer you to our recent science article, perspective article, in which we introduced the term DNA mechanotechnology to describe DNA nanotechnology devices that are used to generate, transmit, and sense mechanical forces. Thank you very much.